How's it going, everyone? It's Chow here, and today we are going to talk a little bit about cell signaling, so let's get started. All right, so signals are basically nothing more than chemical or physical stimuli from the environment. They can come from outside of the cell, like in the case of photons or pressure against your skin, or they can come from neighboring cells, so things like neurotransmitters. But when it comes to signals, we must have cells that express certain specific cellular receptors to pick up and detect that signal. And ultimately, this combination of signals as well as receptors will induce a signal transduction pathway, which is nothing more than a sequence of molecular events and chemical reactions that induce a response to that particular signal. So in the most basic form, we have something like this, right? We have a signaling molecule, in this case, our ligand, that binds to a receptor, and that receptor is going to induce a transduction cascade that involves multiple relay molecules to create some sort of a cellular response. A good example of this is the insulin signaling pathway, which, as far as I know, you don't have to know these specifics because I think it's a little bit too much, but the binding of the insulin, which in this case is our ligand or our signaling molecule, is going to induce a number of different pathways that's going to ultimately, affect transcription of certain enzymes, certain proteins, as well as the induction of GLUT4 molecules to the surface. And these channels, GLUT4 channels, are going to have increased abilities to allow glucose to flow through them and enter into the cell, and the glucose can be used or stored depending on the situation. So GLUT4 occurs in your adipose tissues as well as your muscle tissues. The idea, though, is that you have a signal molecule binding to a receptor, and that receptor is going to trigger a series of events that are ultimately going to lead to changes. So some of these changes, like enzyme activation, cellular movements, they're going to be short-term, but other changes like altering DNA transcription, that's going to be considered long-term. Now, when it comes to the different types of signals, there's kind of four that you sort of have to memorize. You have autocrine signaling. Autocrine signaling is the cell producing some sort of a chemical, a molecule, neurotransmitter, interleukin, cytokine, and it's going to auto-regulate. So it produces that chemical, that signal, and is going to bind to itself and auto-regulate itself. That's in contrast to juxtacrine, which is kind of cellular touching in a way, where you have activation of nearby cells that are right next to the original cell that's producing the signal. And then you also have what's known as paracrine, which is just slightly farther away. So signaling that affects cells that are quote unquote nearby via the transfer of that molecule through a diffusion pathway. And then finally, you have what's known as hormonal signaling or exocrine signaling. And this involves the production of some sort of a signal that's going to pass through the bloodstream to target distant sites. So you can think of it maybe as like in your pituitary glands that are going to be releasing certain types of hormones, but they have to get into your bloodstream and then flow to distant sites and activate things like your adrenal glands. So those are different ways that signals can target cells. Now, when it comes to the binding of the signaling molecule to the cell, or the induction of that signaling molecule to different cells, the important part is this thing called crosstalk. So crosstalk is when you have signal transduction pathways that are somehow interrelated, as shown here. You have certain pathways that are branching. So one molecule is able to activate multiple different pathways. By contrast, you can have situations where you have multiple pathways converging on a single molecule. So it's basically a situation where you have different types of relay molecules that are targeting different relay molecules, but in this case, it's different types of relaying molecules target one specific type of relaying molecule. So I hope that makes sense. And then finally, there are, of course, situations that have to make things a little bit complicated, where you have one pathway that might lead to activation, but that same particular type of relay molecule might lead to inhibition of another type of relay molecule. So this entire idea of crosstalk and the interbalance, this balance between the crosstalks, will ultimately lead to activations and inactivations that will trigger or inhibit some sort of a response. So in order to induce that signal, you have to actually have receptors to detect that signal, right? There are many different types of receptors. Some of them are found on the cell surface, so you have plasma membrane receptors, and these tend to bind larger or polar ligands that can't pass through the membrane easily. So you have certain types of like neurotransmitters or certain types of ligands that are either polar or very big in size, and remember that polar, huge molecules don't easily diffuse across the membrane. So these receptors, such as ion channels, protein kinase receptors, and G-protein link receptors, which we'll briefly talk about in the next couple of slides, these are often plasma membrane receptors that are binding molecules on the outside of the cells. There are also cytoplasmic receptors that bind ligands that are generally smaller or nonpolar that can diffuse across the cell membrane. And an example of this is the estrogen receptor, which is found in the cytoplasm. So estrogen is able to diffuse across the cellular membrane. It's going to bind to the estrogen receptors, and then they're going to dimerize, and then they'll move into the cellular nucleus. And when they're inside the nucleus, they can affect transcription activity, sometimes upregulating it, sometimes downregulating it. So just different kind of classification of receptors to keep in mind. So we're going to talk about some of these receptors. And the first thing that we really need to discuss, though, now, when it comes to the receptors themselves, you kind of have to break them apart, right? You have this relationship between the receptor and the ligand. The ligand, of course, is our signaling molecule, and the receptor is the thing that binds our ligands. The binding of the ligand to the receptor causes some sort of a conformational shape change, and that's going to be what induces, right, that signal to be passed on. Generally speaking, the binding is reversible. There are some situations where it isn't, but we're not going to go into the details of that. So we're going to just go ahead and run through some of these different types of receptors that are going to be binding our signaling molecules and inducing the response. One type, of course, is our good old friend, the ion channel, which allows ions to move into and out of the cell. These ligands tend to be hormones, neurotransmitters, or changes in charge. And an example is where you have acetylcholine, which binds to the receptor and it opens, it's basically an Na channel, so it's a sodium channel. And by binding to the receptor, the ion channel, the acetylcholine opens the channel, and that allows sodium to move into the cell. So ion channel is pretty basic, ligand binding, opening up that channel, allowing for ions to flow through. You also have what's known as the protein kinase receptors, and protein kinase receptors, they're able to catalyze a series of phosphorylation events. So the binding of a ligand, for example, to protein kinase receptors, they often cause the dimerization of protein kinase receptors, and that allows for a catalyzation of a series of phosphorylation events. So phosphorylation, if you think back, is basically sticking some sort of a phosphate group onto a molecule. Usually that phosphate group is derived from ATP, so it's definitely energy driven. And you also have situations where you can remove that, right? So that's dephosphorylation, where you're taking that phosphate group that's attached to a molecule and you're removing it. Just to keep things into track, usually kinases are the ones that phosphorylate and stick phosphate groups onto molecules. And then you have phosphatases that are the ones that are removing the phosphate group. So something to keep in mind. Now, aside from our ion channels, as well as your protein kinase receptors, there are also G-protein linked receptors, or GPCRs. And G-protein linked receptors, they actually do a little bit different of a signaling method. So you have some sort of a receptor, in this case, a G-protein linked receptor. The binding of the molecule is actually going to activate the G-protein. So the G-protein is actually a mobile membrane protein with 3 different subunits. It's normally in an inactive form when it's bound to GDP, but the binding of a signaling molecule to the G-protein coupled receptor is going to cause a conformational shape change of the receptor, which allows for that GDP to be swapped for a GTP. The binding of the GTP actually activates the G-protein itself, and it's going to cause the G-protein to perhaps dissociate in some cases, and certain subunits will activate certain pathways. And by binding to certain activated, certain effector proteins, that's going to cause the activation of those receptor proteins, which is going to induce a cellular response. So generally speaking, when it comes to G proteins, it's inactive when it's in the GDP form, and it's bound to GDP, and it's going to become active when it's going to be bound to GTP. You actually have situations where the G protein actually is able to remove and hydrolyze the GTP back into a GDP form, and in doing so, that's going to allow for the G protein subunit to reassociate with the other subunits and basically go back to an inactive form. But what's kind of fascinating is that it's not really effective at doing that. So sometimes you have to have other enzymes to come in to help, help it hydrolyze that GTP back into the GDP. So you have certain issues like cancers where you have constant binding of GTP to the G protein, and in doing so, that G protein is going to be constantly active and constantly creating certain cellular responses, and that can lead to certain diseases like cancers. So that's something to always keep in mind. And here's just a nice overview of different types of membrane receptors. So you have ion channels, right, that we talked about, and then you have protein kinase receptors. Remember that dimerization, the binding of the molecules, and then dimerization triggering some sort of response. And then of course your G protein linked receptors, where you have a binding of a molecule that ultimately leads to the swapping of GDP for GTP. And then that subunit that's activated will induce some sort of an effector protein cascade. Often things like adenylyl cyclase, convert your ATP to cyclic AMP, which is a secondary messenger. And that's something we'll talk about in probably a following video. We're going to break this into two parts because I think this is going to be a pretty long section. Now, this is the part that always annoys the heck out of people because it's what we have to sort of keep in mind when we talk about biochemistry and proteins and enzyme substrate relationships, and in this case receptor ligand relationships. So receptors, which is denoted by R, bind to their ligands, denoted by L, according to the law of mass action. So in this particular case, we're looking at dissociation constants where the R and the L, this is the bound form, and in this case, this is the dissociated or non-bound form. The unbound form and the bound form will actually allow you to figure out something called the dissociation constant. So this is an important component of receptor ligand binding. It tells you how tightly is it able to bind. So it's looking at binding affinity, which in this case is measured by our constant Kd. So this is the dissociation constant Kd. So Kd is that equilibrium constant for the dissociation reaction, which is represented by the receptors in the unbound form and the ligands in the unbound form in the numerator, divided by the receptor ligand bound form, which is in the denominator. And these are all concentrations. So concentration of free R receptors, concentration of free L, the ligand, divided by the concentration of when the receptor and the ligand are bound together. And so if you think about it and let that math run through your head, the lower the Kd, the stronger the binding affinity. So the lower the Kd, it means that this value here is lower, so more of this receptor and ligand are bound together, so you're going to have stronger binding affinity. By contrast, if you have a higher Kd, it means you have more stuff here in the unbound form and less stuff here in the bound form. So that's something to always keep in mind. Lower the Kd, the stronger the binding affinity. And this will help you a little bit when you're going through your biochemistry classes as well. Okay, so signal transduction pathways then allow you to move forward and initiate some sort of a response. And that signal, that ligand, is going to cause a change in the conformation of the receptor, right? And then that conformational change ultimately creates new activity in the receptor, and that new activity results in activation of other proteins, right, in that particular cascade, and that's going to lead to a response from that original signal, which again, that original signal is like a ligand or pressure or changes in charge, something like that. So ultimately, we're all back to this particular crazy little formula over here, where you have some sort of a signaling molecule that's binding to a receptor. inducing transduction of various relay molecules that's ultimately going to affect cellular responses. So this is the first part of the video. I want you to go ahead and pause this or end this and think about it. And then once we get through that, we'll move on to the second part. So I'll see you in the second part of this signaling transduction video.